Copy.
Okay, so they're on the chat, not on the line right now. We also have... Yep, go ahead, Casey.
Usually folks from the Interspace Center don't join on the telecon, but the Interspace Center is located at the University of Rhode Island, and they are a key partner for all of our Okeanos Explorer missions as they help facilitate our communications and broadcasting our live video feeds to the world.
And we also have two participants who are probably not on the telephone line, but who are active participants in our chat room. Asako Matsumoto from Japan, and Tina Molitsova from the Shirshov Institute in Moscow. And for both of them, I think it's, well, for Tina it's quite late, and for Asako it's quite early.
Quite early,
so, yeah. So we appreciate the active participation of our colleagues and team members from
all over the world.
Okay.
Yeah. What we're doing right now, the pilots are using the color bar and the black and white card that you see on the screen to get color balance and to maximize the lights, to bring you this incredibly wonderful high-definition video that you'll be seeing throughout the dive.
Like
they take a little while to position
right,
the remotely operated vehicle, the D2, on the seafloor so that we can make tracks across the seafloor. And you can see the vehicle if you turn to the video channel that has the Sirius view on it. And we're sitting at the top of, it looks like a bit of a scarp. And on the top of that, there are a whole bunch of white patches that I'm sure are biological.
Maybe so.
Oh, it looks like there may be some corals. I see. Are there any sponges? Shirley is a sponge expert.
Yeah, I'm thinking. Yes, I'm saying.
And she goes nuts.
I'm saying corals, and I am seeing some sponges.
She
as well.
is. Oh boy, this is going to be fun.
It's going to be fun. Yeah. So we've done a series of dives now along some seamounts, and honestly, each one has been
incredibly unexpected. I'll tell you,
different, unexpected.
Yeah.
So I'm really curious to see what the geology and the biology is like on this particular reef.
What we do in order to plan the dive is we have bathymetry, that is topography of the seafloor, that's collected with sound, and we have found that sometimes the sound source gets a little confused and gives us erroneous peaks and erroneous pits in the seafloor, and so when we get down there, then we can see the detail. And what we found thus far is that despite the fact that the bathymetry is really good, I mean, for the fact that we're collecting it from depths from, several hundred meters to 6,000 meters deep, gives us a good feel for the general topography. But when you get down on the seafloor, there's a lot of detail that's missed by that type of survey. So here we are sitting on this, the edge of this scarp that we're going to be traversing throughout the dive. And already we're seeing detail that we hadn't expected to see down here. We were actually hoping, and one of the objectives of this dive today is to try to collect real fresh lava.
And why would we want that?
75.
We would want that because when a lava flow comes out of the ground, it starts cooling right away. And when it starts cooling, the crystals that form the composition of that liquid start to crystallize out of the liquid at various temperatures as it cools. So what we really want is to find out what the original liquid composition is. And that is captured in the glass that forms the minute the lava touches the seawater and freezes.
And another important objective of these dives, especially in the shallower areas, is to document the occurrence of precious corals and commercially important bottom fisheries.
Yeah, right.
Because that's part of the management responsibility. And
of the monument,
yeah, right, the monument.
Do you want tops out too?
And there's a shark. All right, it is a good day. We start out with the predators.
right, deploying
Yeah.
upper swing arms.
Bruce Mundy called that a silver tip shark.
Oh, silver tip. It really was a pretty
Silver tip,
guy.
It was really pretty. Yeah, really pretty. Okay.
I can see the broken branches of corals littering the seafloor, too.
Go ahead, watch lead. We are ready. Yeah, sure. Let me just stabilize here a little bit.
So the other rounding out
All right,
our
pilot, your light
team
bars are
are
out.
Okay,
the
copy that.
Ramping
men
down
and women
HQ.
who are part of the ROV
HQ
operations, and they are from another OER, Ocean Exploration and
Center
Research
your-
partner, the Global Foundation for Ocean Exploration. So they're going to introduce themselves right now.
Kia ora, everyone. Good morning. Welcome to the New Zealand time zone. My name is Jeff Williams. I'm your pilot for the morning. And to my left is our navigator.
Right? Mike's.
There we go.
Hi.
Here's our navigator.
I'm Richard Stoner, and I'm the navigator for the first shift this morning.
And I'm Sean Kennison, sitting co-pilot this morning.
And all the way over in the corner is our videographer.
You can just see a silhouette in the corner.
And
Good morning, I'm Tara. I'm the video clipper today.
And our videographer forgot to tell us who he is, but his name is Roland.
where's is our dive super on the line? I know he's not in the room, but he's on the line somewhere.
No, but we do have our coordinator.
And I just want to say that we're all from the Global Foundation
Yep.
for Ocean Exploration.
Great. Thank you very much, ladies and gentlemen. I think we're ready to start diving.
Watch lead, pilot. As you can see, we're just here at some interesting geology. Is there any interest in a rock this morning?
We are going to, I know we're interested in some new rock, right?
Right. Well, as I mentioned earlier,
Yeah, go ahead.
Shirley, you were busy, my dear.
Yeah.
Shirley and I are really good buddies. We've known one another for years. Yeah, we're looking for fresh rocks. So these rocks look brown, and that generally means that they have altered in contact with seawater. So these guys are kind of old.
Okay.
What we're hoping to see is some rock that looks as if it has a shiny glass surface. Often when lava is erupted on the seafloor, it sort of squeezes out of openings on the seafloor the way
lower center.
toothpaste squeezes out of a tube.
There's a brown and a
So
black.
it actually looks like a tube of lava coming out of the seafloor, and that's what we call pillow basalt. The rim of that, or the outside of the tube, gets frozen almost immediately because the ocean water has such a high heat capacity that even just, a few centimeters away from the surface of a lava that can be 1100 degrees Celsius, the water will be like 3 degrees Celsius. It's just amazing. So if we, during this expedition, come across hot hydrothermal vents, you may see animals that are swimming around right near a black smoker that it's pumping out fluids that might be several hundred degrees Celsius, and they get up within a centimeter of the plume.
What's that hiding in that crevice there? Is that
I
a
think
crab?
that's
Looks like a
crab,
crab
or
hanging.
not a squat lobster,
No,
huh?
it's hanging. No, it looks
Too,
like
it's,
it looks like a crab in
its
there.
nippers are too big.
Yep. Yep.
I
Okay,
think
roll in.
it could be
Easier
a
to
squat
roll with it.
lobster, actually, because they can have pretty long pincers like that.
Okay, stand corrected. That's the beauty of having an expert on shore.
Yep. And if we just look up, yep, you got it.
What are these? Let's see.
They look like they are, just go over
A
little
bit to the right.
And these
what
look
is these
like
guys?
they,
Excellent.
yeah.
What are
It
they?
looks- They look like they're sponges, probably lithistid sponges. So there are some sponges that have a lot of sponges have microscopic skeletal elements. But the lithistid sponges is a group of sponges for which these little spicules or microscopic skeletal elements interlock to form a very rock-like, really hard, dense consistency. And we often see them at these depths. Okay.
And as we back out and look at these rocks, like I was saying earlier, I think these guys are fairly altered. It's interesting, though, that they look like maybe they might have kind of fit together.
Rolly, I'm going to go up where it's a little
when
better
they
terrain
were
for me to-
originally deposited on the seafloor and then broke apart.
Should also spiral around to look at
Most of the volcanoes out here in the Marianas are. Yep. I hear somebody on
Go ahead,
shore.
Mary Jo.
Go ahead.
It was me, Patty. I was wondering what you think about the way the top of the bridge here is breaking these like platy structures kind of off to the right, and if you think maybe it should be like an ash or a fine-grained volcanoclastic layer.
Yeah, I think so too. What we're seeing looks so layered, and all of the broken pieces sitting down the lower part of your screen, probably detached from some of those layers a little bit further up. And I'll bet that is ash. Sometimes volcanologists can date an eruption by looking at the ash and getting potassium argon or other types of radiometric age dating from the materials. And there's a blob. Is that a blob?
Oh, I think it might be a sponge blob.
A bioblob. A bioblob of Shirley's favorite type.
I have a lot of current, so I'm just going to handle the joysticks if you want to tilt my camera.
Absolutely.
Sean,
Okay, so this is
Yep.
another sponge. This is very different from the sponge that we
just
saw. Yeah. Interestingly enough, it's in the same kind of general grouping. It's a demo sponge. It has spicules made out of silica. It's even in the same, so that's a class of sponges, the demo spongy. It's even in the same order of astrophorid sponges that those little lithistid rock-like sponges were, but this one is quite different.
And you can see the hair-like extensions from the body.
Those hairs are spicules. They're made out of silica.
And they are sharp.
They are sharp, yep. Beautiful. And you can also see in this image the pores on the surface of the sponge. So sponges are in the phylum Porifera, pore bearer.
And there's a little tiny something crawling around on it.
Yep.
Is that a shrimp?
Yeah, looks like a shrimp. Looks like a shrimp.
Okay, guys on shore, what do you think that little guy is? I think
video. Can I
Tina Sova says shrimp. Thank you, Tina.
You got it?
A little slight tilt down.
Slight tilt down.
I think after that I see a
Shirley, so as far as you can go on the sponge is astrophorid, is that correct?
Actually, I think it might be in the Pacastrellidae, Chris.
Okay, I'll go ahead and punch that in the event log.
Yeah, okay.
Top right.
I don't know if you noticed, Chris, but the spicule brushes were kind of arranged kind of a circular area or circular fashion, so that would tell us something a little bit more diagnostic. It might be Characella or something like that. I'll have to look it up.
And as we look at the geology right above the sponge, the pores or holes in that material, it certainly looks like ash, and there's a little white blob of something way over on the right.
Oh, that is probably another lithistid sponge.
Far right. Okay.
Yeah, that's probably another lithistid sponge. So these types of sponges are fairly common at these depths.
And the white thread-like things you see there are
probably
tube worms, right?
Yep.
So Shirley, why would a sponge put itself inside a rock? I mean, how's it going to get stuff to filter out?
Oh, there's all kinds of water that's circulating through there. So, and they can set up their own water currents as well.
Really?
Okay, Scott wanted to know what the blue blue thing was on the brown rock behind the sponge. So yeah, I think we're away from that now. Small.
Push this off.
Oh, I see. Yeah, I see it now in the instant replay. You know what, Scott? I think that might have been another sponge, but I'm not sure.
It's kind of difficult for us to...
I just noted it because the color is pretty different from most of the things we see down here, but sponge certainly seems reasonable. It's awfully small.
Yeah, you know what? There's another blue that I've seen. It's like something like dark blue like that I've seen in the Atlantic, and there are little crinoids that we've seen from time to time. Crinoids. But this did not look like what we see over in the Atlantic. I think it's a sponge.
Tilt that camera up for me, please.
Scott, if you recall, the other day we saw a larger blue sponge as well, and looked like it might be a new record.
Well, Shirley, it looks like a lot of these things
Should we look at one of those large
are,
things on
golly,
surface?
you know, I'm not going to take a guess.
Yeah,
I was
do that.
thinking
that
they might be
should.
corals,
At this angle,
but
we should
they
look at
look,
one of these edges
some
of
of
the
them
rock.
look like sponges.
Yeah, they do.
And this
Just to get
terrain
the edge
looks,
on view.
this looks pretty
Yeah.
old.
I have to get down and get a toehold. The current's
Relatively
really
speaking.
strong, and
Not
it
billions of
takes
years,
me and the tether, and it
but
keeps bouncing me around.
tens of
so
thousands to hundreds, I don't know.
settle
It's
in
pretty
here.
old. It's all altered.
There's also
Brown
a
color generally
biological
means it's been
purple
altered by
Okay,
seawater
Sean, you've got my camera again.
contact.
I have your
wow.
camera.
Wow, what
That-
is this guy?
Another sponge.
Is it really?
Yeah.
Oh, yeah, I can
We
see
were just
all the
gonna
spicules.
We were going to show you guys the geological
Okay.
layers here.
Sounds good. So, yep, pilot is maneuvering the ROV. There's a fair amount of current, so he's trying to get a toehold into that current, get it positioned so we can be really still, so our videographer can get some good imagery. It looks like in that crevice, let me see.
Really,
I don't know if
don't
that's
think
a
they got your
hydroid.
I
message.
noticed as we were zooming in that on the second sponge, it looked like there was something flexible and brown on the top of it. Maybe we could have a look as we're leaving.
Sure.
Okay.
All
So
right,
tilt straight up.
tilting up.
We'll get the thing on this and the thing behind it.
Very
after we get this
interesting
guy.
sponge.
Yeah.
It looks
Copy
like
that.
it's got, it's picked up.
Yeah, so these spicule brushes,
Debris.
the spicule brushes are traps for sediment. This is kind of unusual because I'm thinking it might be
you can
another
see the current's kinda taking
lithisted
the tether
one.
pretty good,
Perhaps
too.
it's called
Yeah.
Coralistes.
It came
Yes.
down some, but
Even when
It
I'm
might still
stable, the
pull
tether
specimen.
just keeps
yeah.
Do we have lasers on?
wobbling. me.
Lasers are
And how
off.
about the
I
one
will
at the
turn
top of
that
the screen?
on.
Well, and that's
Is that the same?
another
Now?
a different
Lasers on?
species. So we're seeing a lot of diversity in the sponge fauna
Wow.
here already. Yeah, haven't
Okay,
even
we'll
moved that
pull
far.
For lasers, and we'll look at the purple thing at the top
Scott
off the
said
screen.
there's a
Purple
brown,
thing,
something
top
brown
off
on
screen,
this.
Copy.
Oh,
Yes,
okay. Who
there we
knows
go
what that
again.
is? Who knows what that
Oh,
is?
That's an
We know what that is.
anemone.
And what kind?
Oh, I can't
Lasers
remember
on
what
it, video?
kind.
Pom-pom.
Yeah.
Oh, of course I should remember.
I know. Don't even get into that
No,
discussion.
we will not get into that discussion
The
again.
cheerleader discussion.
Oh, shush.
Oh, this is very, very cool.
But you
So
guys,
yeah,
I'm sure you can probably
Rotate
tell.
left.
Maybe.
We are excitable types. This is a beautiful animal. We actually were able to collect one of these animals a couple dives ago.
Yeah, our ace pilots really
Oh, man.
carefully went under that. These are really the pom-pom anemones are actually, I did a little bit of literature research on this.
Good for
They're
you.
really loosely attached to the bottom, so they can actually roll along or tumble along the bottom as well. And all of those frilly things that we're seeing in the background, the white frilly
go for it.
things,
Those are sponges.
Those are sponges. Those are lithisted.
Yeah, lasers touched it briefly, so it's
lithisted
about 10
sponges.
centimeters.
Yeah.
Exactly.
Yeah, Scott France typed into the chat room that they're like tumbleweeds. They
Slight
don't
tilt up.
attach hard to the substrate, but they're really fragile too. And so once you start, handling them, all of those little tentacles start falling
Start to
apart.
drift,
But our
trying to keep the
Crackerjack
full
pilots the other day were amazing at collecting this. Came up in one of our bio boxes, almost intact. Just a few of the tentacles had sloughed off, and yeah, it was incredible how well the still intact.
Yeah, the pilot had a little bit of trouble with some of the tentacular tissue that remained stuck to the arm of the manipulator. It's kind of difficult, to flick your arm if you're a robot.
All right, pilot, I will give.
And he managed eventually to get it off.
Yeah, the tentacles, these types of anemones are referred to as deciduous. So just like trees drop their leaves, these anemones drop their tentacles.
But look at all those
really
sponges.
abundant, really abundant.
One up on the top of the ridge there, Roy?
Yes.
All right.
All right, pilot, I'll have to rotate with you, but I don't think that will be an issue.
All right. And then our direction of move is like 160. Is that right now? Nav.
And here is one of those
160,
yellow
is that gonna
sponges
be our
that
direction of
is
move?
a.
Our direction will
Oh
be
boy. Oh boy. Patty can't remember the name.
Oh, okay.
Sponges are corals.
Copy
Corals.
Yeah.
Gosh. Yeah, I can't even remember the type of animal. The little yellow thing on the bottom of the screen is a coral.
Yep. And is this one of the ones that was?
We've collected one of
Was
these.
this Agucha samia,
It's really
Chris?
hard to hold.
Yes, so
Okay.
I saw one when we first
You
dropped
got
down, and
camera.
I went ahead and put it in the event log. You caught me looking at that anemone. I was trying to remember what family the Velocira is in, but the pom-pom anemones are in the Liponematidae. And
What can you tell us about the coral?
Any of the Liponematidae seen in Hawaii. So I don't know if it's in that family, and I'm trying to figure out what family the genus Velocira is in. That's another possibility. But when you collected one, I must have been gone. I was gone an hour and a half one day. Did anyone come up with an ID on that one?
No. Tell us about this coral.
Okay, well, this is a hard coral. It's called a scleractinian. It's related to all of those shallow, big, massive corals on coral reefs, or the plate corals you also see on coral reefs. It secretes a very hard skeleton. You can kind of see a bit of that skeleton right at the base in the lower right.
And a baby on
It's
it.
a very unusual one in that it forms either kind of very small little aggregations of polyps and little groupings or colonies on the bottom with this brilliant yellow color. I don't know why they have that marvelous color, but really interesting. It's known from these depths. It was clearly named after somebody. Its genus name is Aguchisamnia. And
all right, let's go full wide.
Either Aguchisamnia fistula or Aguchisamnia serpentina or something completely different.
Pilot, do you
So I
want to
think
slide
Chris,
tilt up
I
before
think
get any
that
cameras?
Steve Cairns named this, and he
Right up
named
to
it
the,
after someone
edge of the skids.
who worked on
right.
deep water corals, and his name was Oguchi. His last name was Oguchi.
All right.
Yeah, that's
Let's
what
get a
I
look
think
uphill
also.
here and get set up for our first move.
Science,
Copy
is there
that.
anything else? I believe we imaged each
I kind of stretched
species
out in
we've
front of
seen
you.
here, but if there's something you guys haven't seen,
A little bit. So I have you
or would like a
45
better
my
look at,
camera.
let us know.
Oh, okay.
So you're about a nice
Yeah,
spot.
Roger that. Just some maybe,
pull up further,
maybe
we'll
just
see what
one
we
more
want.
closer image of these frilly sponges, please.
What? One more image of a sponge.
Yeah, these frilly, wavy sponges.
That one that's left of lasers, the big one, would be a good candidate.
Gonna pick a different one.
You're headed for an ugly one.
Well, ugly is a human judgment, so sponge doesn't care.
Mic's not open.
Oh, wait, you're on mute.
All right.
These are sitting
All
on
right, pilot, I
ash
will take your
deposits
camera.
that probably
Okay,
have
go for
hardened
it.
over time
All right.
by being cemented,
I have your camera.
probably by alteration.
Tilting down.
You can see I'm not very well settled on this stuff.
So my colleague, Michelle Kelly from NIWA in New Zealand, has done a lot of work on these deep water lithistids. So we'll go ahead and get some image of these, and I'll send them off to Michelle, and I'm sure she'll be able to identify them. But my guess is that they are in the family Corallistidae.
As good as it's going to get.
Going down. Stopped.
Again, these are demo sponges, and but they're very. Oh, look at that! Nice little hermit crab on the top as well, and some hydroids over to the left. Some worm tubes. So you can see there are a lot of other animals that attach themselves to the surfaces of many of these sponges.
Okay.
Those lasers that you're seeing in the screen are 10 centimeters apart, or about 4 inches apart. So it'll give you an idea of scale. Great, thanks, pilot. So you can see that the most dominant organism at this depth is this lithistid sponge. And again, I'm pretty sure we'll be able to put a genus and species name on this. Talk to my colleague Michelle Kelly in New Zealand.
Okay. Well, let's see, we're trying to go due south, but the ridge is saying, what, 160 did you have before?
Let me turn into it, see what it...
Yeah, I'll do a slight rotate port.
Kind of feels like 150 for me right now.
150.
150 seems to be straight up the hill.
Okay, from your position.
So if I turn 180,
yeah, I think
that's going across the ridge. See that? That's 180. So I think we want to go 150.
Yeah, that looks pretty good on Sirius sonar.
Okay.
I am fine. Do you want to try to lateral over a little?
To just get in the center of your view?
Yeah.
I'd be happy to do that.
Because I'm at about 165 right now.
All right.
So a little lateral, and we can center up.
It looks like a 40-meter move in that direction will give you about a 15-meter inclination.
Oh, that'd be great.
A little scorpion fish under that ledge. Some more of the cup coral over to the right.
Fish went off right.
Not sure what these branches are.
Oh, there's an interesting, different sort of critter there.
What are you seeing?
Yellowish.
Yep, up and over at the 9 o'clock position.
Yep.
Yeah, okay.
Coming into the lasers.
Okay.
Out there. Maybe it's the same sponge.
Not sure. Let's take a look.
Coloration.
Yeah, it does.
All right, pilot, that looks pretty good.
Our ROV team is really amazing. I mean, they do this a lot. But, Patty and I just come out here, occasionally, but the ROV crew is out here. They're seeing a lot of, they're on every mission. They're seeing a lot of different things, and often they'll spot something.
Yeah, they're a lot quicker.
Okay,
a lot quicker than we are.
Take a zoom on that?
Of course, they have the really big screens.
Oh, really nice. Oh, my gosh. Oh,
Sorry,
my
pilot,
gosh.
did you say take camera?
Yeah, go ahead, take camera.
Beautiful.
So there's an urchin, there are
Rotating
brittle
down.
stars. It looks like there may be
Still
a
settling
hermit
in, but
crab.
that's all right.
What can you tell?
Don't
Oh,
zoom
and a
any
slit
further.
shell. Oh, no, wait. It's
Yeah,
not.
camera's,
Well, it's a slit shell, but it's got,
Yeah,
it's got a hermit crab in it.
Hermit crabs borrowed it.
Yeah. Scott, can you tell us anything about this coral?
Oh,
Chris is saying it's an
Without
analopsamia.
facing into the current, because that'll put me in a
Okay,
better
so
position.
what's
Yeah, that was going to
what's
be my
sitting up
answer.
on the?
Wait.
Scleractinian coral, which is a stony coral.
Your
Those are the
white
types of
light
corals
is
I think that most
really
people would be
flopping
familiar
lower.
with that make up the
Yeah, I
shallow
noticed that
water
earlier. I
coral
think it's
reefs.
just in that position.
But in
It's
colonial
getting some
form,
sort of
as this
maybe a
one is,
vortex or something from the
relatively
current.
rare in
Because
the
when it
deep.
swaps, the
I'm not
opposite light
too
does it.
familiar with all the different genera, but Analopsamia is one that's fairly common in both the Atlantic and the Pacific and has
Any
this general
chance of
growth
getting it
form.
from the right-hand side?
No.
There are some commensal organisms,
I
and
have to have both
I'm
toes on the
wondering
ground
if
in
I'm
this current.
seeing a hermit crab in another
Yeah.
shell crawling on the
We're not
top,
gonna be able
Yep.
to see
Yep,
the
that's-
polyps.
and a brittle star further
Further
on
up.
The on the other
and
side.
an urchin.
A condominium.
Yeah, these little oases of activity.
Absolutely.
One of the things you can see on this coral is that all the feeding polyps are on one side, which is a reflection of the dominant current. And somebody else can chime in here, but I think it may be that the current is coming on the side where there are no polyps. It creates eddies as the current moves through, so that the water sort of gets turbulent and slowed down, and prey items in the water can get captured by the polyps that way. At least that's my interpretation.
Oh, that's very cool.
That makes sense.
Yeah, makes a lot of sense.
Hey, watch Lee, I confirm the direction of the current movement.
Yeah, so, we've got so many sponges and the coral and everything living on this ash bed.
Yeah, go ahead.
And it's got to have been there for a long time.
Yeah. Oh, and there's a barnacle.
Look at its little feelers coming out.
What are those feelers? No, come on.
No.
Cirri.
Rats. I'm going to write that down.
We're teaching each other.
How do you spell cirri?
C-I-R-R-I.
Right.
All right, pilot.
Oh,
now we'll
another
give
urchin
camera
in the
back.
background.
Yep. And I think we can start that move.
Yep.
Okay, do we confirm?
Okay, so I'm going to take the contrary point of view
yes,
here.
5-0.
Given that most of these deeper water currents are tidally driven, and the fact that we've seen complete
30
opposite
meters.
changes of current on some of the dives that we've done, I don't see how one single orientation or one single face of that coral could be the down current
Bridge
side.
from ROV.
I love it.
And this is what's really interesting about scientific inquiry. We can debate, we can test these
Who is this
hypotheses.
right here coming into center
Oh,
screen?
what is that over on the left? Is that a siphonophore?
Really?
No,
Do you want
what
me to
is
take
that?
camera?
Bridge from
Is that a
ROV nav.
siphonophore?
Go ahead,
It's
Nav.
just,
Like in the water.
oh, like
Could we
looks
have
like
our
almost
first
like a little
move,
pogo
please?
stick going across. Oh.
Requesting a 30 meter
Snap
move,
zoom roll.
heading 150.
Great, thank you.
Speed
We
0.2
have
Lots
knots.
of them
an urchin.
Yeah,
Right.
minefield.
That must have been a siphonophore.
Yeah, that's what I'm thinking too. It was being
Good copy.
carried across our screen pretty quickly by that current.
Yeah, and I'll just notice in response to Chris's comment, which is perfectly
Now,
valid,
bridge,
that siphonophore and most of the particles are being carried from left
to right.
which is the orientation that I was referring to
Thank
of
you,
that
Rich.
coral. So you may be completely right that tidal currents may switch it around, and perhaps the coral will mostly feed when the currents are moving one way or not another. I don't know. It's just speculation. But right now, I do see the current seems to be moving in the orientation that it would be eddies for the feeding.
Right, and maybe they could use two strategies. So, feed in when the current is coming from either direction, but take advantage of and take advantage of both. So
Maybe there's a predominant one, though.
yeah.
Absolutely, and it's just an interesting question because not all corals take on that shape. Some have polyps on all sides. There may be various reasons for the
Are we underway?
distribution of polyps on a
I
colony.
believe the ship is moving.
Okay, great.
Yeah, we are underway,
And
correct?
for those of you
Control.
looking at
I
the
don't
video
see
at
any movements
home,
quite yet,
you
but
see
I
two
just saw
tiny
the ship
red
move.
dots in the center of the screen, and
Yeah, it
those
looks like
are
it's,
from our
7
lasers,
minutes.
and those dots are
I'm
10
not gonna
centimeters
get stretched
apart.
out too far.
So
there.
when we get close to the seafloor, you'll see them a
I
little
do about
better,
40
but
my
that's about
camera.
4 inches. So we
Hey,
use that for scale.
You want to stow that starboard?
And also,
starboard
if you're
lower,
watching
or
from
at least
from
rotate it
home,
back a little bit or
our
something.
Rotating back.
citizen
Copy
scientists
that.
watching from home, you can switch
starboard.
feeds. I think there are
back.
3 different screens that you can toggle
right.
back and forth on, and you can look at the image that's coming from the Sirius, and you can look down on
maybe
the
it's
Deep
back there,
Discoverer,
we'll get-
and you can also then get an
We'll try up
overview
and
of what
down.
the bottom
Oh,
looks
that's
like,
great.
and all those
Yeah,
white
just try
things
to
that you're seeing on the bottom are
Do
those
something to get
lithisted
it out of
sponges.
whatever eddy it was in there.
Now I'm
Yeah.
seeing now in the image
I'll keep an eye on it.
like some
See if it gets
small,
better.
well, yeah, but I'm seeing some smaller white things that I didn't happen to notice before.
Sailing valve.
You're
Yeah,
talking about
right.
these tallish things?
Well, yes, and also the smaller ones, so there are some white dots on the surface.
Oh, okay.
Yeah.
That's
Yeah,
kinda-
so just in that, just as they lateraled over, the dominant fauna just seemed to change between, from that lithistid, those frilly lithistid sponges, to what we're seeing now. You can look at that if you.
Yeah, right now we're off on the side of the ridge that we're traversing.
And so having them positioned up on that ridge was positioning them so they could maybe take advantage of the particles in the water. So we're seeing another, we're seeing a couple more corals.
You ready?
Do you see any of the white spots that you want to take a look at, like that one lower strips?
Yeah, the one
above your
laying down is
actually the one that's just up above the dots.
Okay.
Pilot.
Yeah. Go ahead and
up
zoom
above
in there,
the
Rolly.
lasers. Right, there you go.
Showing you
Yeah.
by the camera.
Okay.
All right.
Oh, and now we're seeing.
Rotate down.
I think that's a shrimp.
Is that a shrimp in there? Okay, so the little. Oh, look at that.
That's a sponge, Shirley.
It's a squat lobster over to the left, and then the sponges. That sponge is another lithistid sponge, and it's. Oh, look at that beautiful shrimp. So, this is a target-rich area.
Well, anytime we zoom in, it's that way. It's just
Oh my gosh.
unbelievable amount of biomass on the seafloor. Every dive we've had, that's been the same thing. As the pilots get us positioned and the videographer zooms that camera in,
So
it just fills up with life.
So I
We
definitely.
are 25 meters through the
Definitely going to
30-meter
have
move.
to send these
Understood.
images to Michelle Kelly and ask her to help us with some names on these, because I'm quite sure she'll know what they are, because she's done some work in deep water areas around New Zealand.
Small tilt down.
And
A lot of them there.
a lot of them there.
They're kiwi sponges.
Yeah. Oh, and what's that?
Looks like a tree of some kind.
Yeah, it does. Okay, so Mary Jo is asking
Have to
Patty
go full wide, guys.
what you think of the draping
And I
and
need to get
texture of the
Interesting, all the
lithified ash
debris here.
layers.
A lot of
The vugs.
coral
Okay, what's a
debris.
vug? And the surface textures look like an underwater pyroclastic flow deposit, maybe a boil over or a collapse of a submarine column. What do you think about that?
Absolutely.
But
could
first
be.
tell us what a vug is.
Okay, a vug is a
Okay,
hole.
pushing ahead.
Well, I should have said a vug is a puka, and then you'd have to ask me what that was. That's
You mean
a
like
Hawaiian.
the puka necklaces?
Yeah, puka shell necklaces have holes in them. Those are from
This
what are
white.
the little things that close off a
Let's see what this
snail
white is
shell?
here.
Yeah, a perculum.
Yeah, thank you. I can remember. Perculum. Okay.
Yellow
So-
sponge. Yep.
Mary Jo, I think you nailed it.
Rolly, can we take a partial zoom on this?
Wow, this is Sponge City.
Is this
I
like
know.
a
What are those
crusting
yellow-
sponge of
yeah.
some sort?
I don't know what the
I
little
don't know.
yellow guys
The
are,
yellows
but
are
let's take a look at the white incrustation,
Do you want me to
which
get a
looks
camera?
like a really
Let's
bizarre
just,
amoeba
I don't want any more
sponge.
zoom on that.
Okay, copy.
I can't hold steady.
Yeah.
Now,
fine.
yesterday we saw white. Oh,
right,
no, oh
take
my
it out.
gosh, look at that. Yeah, if you can zoom in closer, that would be great. So this is
I did see
Go for it,
an
Rolly, but I
encrusting sponge
Don't know
sort
that I'm
of.
going to be able to hold it.
Yeah, I'm just waiting for you to go back to the.
this is
That
you can actually see the
Oh, the
canal structure.
pores.
Oh, cool.
Oh, that's beautiful.
Oh my gosh. Isn't that pretty? Okay, and so this is actually thinly encrusting
Yep.
over that
Over the
that
ash.
yeah. But you can actually see the pores, the holes, through which water enters and exits the sponge.
Just on the left side of the
Yeah,
screen.
yeah.
Moving
But you
toward
can
the center.
also see kind of like a star-shaped pattern.
You
Those are the
can.
canals through which the water is being pumped. So these sponges have specialized cells called collar cells or choanocytes, and they have little
And they make
flagella.
juice.
No, they have flagella on them, and the flagella set up a water current that pulls the water through the sponge, and in that water are microbes, small particles
See,
that
it's
they
like
feed
built up.
on. And they go in the sponge and throughout the sponge, and then they exit the sponge.
I wish that was too much.
And
Oh, look.
Oh, really cool.
That is.
There's a sea cucumber.
Yeah, Scott says this is the sponge version of a stoloniferous octocoral. Well, I will tell you, in terms of being able to scrape it off of the substrate, you're absolutely right. Nearly impossible, and it's like Asako told us, they don't ever try and scrape things off of the rocks. They just take the rock with the organism on them.
See the clean white spots?
Clean white spots.
A little bit up.
A little bit up. Oh, okay.
So those are just clear. Those are clear patches of the sponge where
it looks like
sediment hasn't been deposited.
I don't think we're moving anymore.
And what is the feature on the right-hand side?
The right looks like a skeleton of a cup coral.
Ah.
So that just fell down slope and landed on top of this poor sponge, huh?
Well, no, it may have been in place like that, and the sponge across. Yeah, it may have been in place.
Video is clear.
Yeah, remember the other day we saw a lot of dead skeletons? Oh, man, there's a yellow patch. What the heck's that?
Okay, let's try to get the yellow patch before I lift
Shirley,
up.
I might have missed it, but did you have a guess as to what this was, what family, what order, or even what genus this sponge could be?
Not a clue. If I had to guess, I would put it in the order Poecilosclerida, Chris. So demo sponge for sure.
Sorry. Nope.
If I had to guess, I'd say Poecilosclerida, but boy, that's just a swag.
Oh, look.
One of those,
Yeah, I know. It's like all over the place. I know.
Oh, my goodness.
Okay.
We got a lot of animals down here, people. I love rocks,
Let's take a look
wow.
at that yellow encrusting.
Yep, going for the yellow encrusting.
Yeah, and there's a fish up above it.
Yeah, I saw that.
Fish.
Kind of like popping up and down off of my chair. So excited. Ew, boy, what is that? That's another, looks like it's another sponge. Yeah, it is. I'm not so sure that's Poecilosclerida, though. That might be more like a dendroceratid, Chris, if you're writing down these scientific names. So, but demo sponges for sure.
Now, there's a little chunk of it over on the right and down to the lower right as well. And
take a look at the water lobsters
hiding underneath one of those flanges of ash.
Continue to tilt up the fish.
Whoops.
And there was a
in this area
ctenophore up there, like we saw yesterday.
Across it with a couple of mounding sponges, but so far we aren't seeing a lot of stuff sticking way up.
Highlight over to the left if you could take a look at the ctenophore when you have a chance. That'd be great.
I think I'd like to add, yeah. You're getting multiple.
Okay,
we're so excited.
Yeah,
if we could just get a closer image, of that, we can get the fish and we can get the sponge.
Slight tilt down.
The yellow sponge?
No, the yellow sponge and the fish. Okay. That's. Yep, there you go. We're dealing with strong currents too.
Strong currents.
In the picture.
So the pilot is maneuvering the ROV.
But he also has to wait for the ship and the Sirius vehicle to get aligned so that we have enough slack to be able to let the ROV move about on the seafloor.
Right, and he wants to get in position and get still in position so that we can get good video, because just like if you were trying to take a picture with your camera, have you ever seen these things called monkey cam where you're just like, going all over the place? That's not what we do. So we get in position.
All right,
and that way our videographers can get outstanding imagery from which we can get our beautiful video highlights as well as the still images. Okay, so we're hearing from Bruce Mundy and Tara Harmer-Luke that this could be a duckbill fish. We saw one of these yesterday, Crionema. Yeah, a different one. Bruce is saying this is a different species than the yellow-green blotched one that we've seen on different on previous dives. So, Bruce, do you think this is different from the one we saw yesterday?
Well, now that we're zooming in, I begin to see these yellow-green blotches. You know, the angle makes them a little difficult to see. So actually, I'll change my statement about that. I think this is the same species that we saw
pull out
before.
a bit, Rolly.
Just couldn't see those yellow-green blotches. So this is I guess we call it the green blotch duckbill fish.
Tilt up a
Now,
little
these are
bit.
pretty common at this depth. They're found
See if I can
throughout
back
the Indo-Pacific Ocean, the Indian and Pacific Oceans, and there are one or two species in the Atlantic as well.
Tilt down.
They're bottom fish, like a lot of the others, like the green eyes. They probably feed on little crustaceans, are probably ambush predators just waiting there, not cruising around too much, maybe moving forward and scooting along the bottom.
Where's the team for?
Things like
Big
little shrimp.
pin on the sponge, upper left. Straight ahead and slightly left.
Copy that. Straight ahead, slightly left. The white sponge?
Yes.
Okay.
It looked like there might have been a limpet nearby as well.
I think barnacle. Was a barnacle.
Barnacle?
Yeah.
Okay.
Okay, so yeah, Watchley
sponge coming to
pilot.
center.
That looks like was one of the. It's one of those benthic ctenophores from yesterday.
The ctenophores we saw yesterday were attaching themselves to rock surfaces.
Instead.
And what
Sean, if you tilt onto it.
Yes.
what Frank was telling us was that
Slight tilt
their
up.
mouths are at the base of the animal, and the tentacles line, if this is the
Maybe
same
better
critter,
off
Slight tilt up.
and it looks like it,
Looks like it has 4 lobes.
or maybe 2 of them.
Oh wow!
Usually they just, the ones we saw yesterday only had 2 large arms
Slight tilt
or
up.
bulbs sticking up from the
Yeah,
base.
you're looking at more than one individual here. They only would have 2 of those tentacle sheaths.
And it's
So are
is
there?
it eating the coral?
Slight tilt down.
Gotta try to come around it here a little bit so we can see the 2 individuals.
So one of the problems with figuring out what group of ctenophore this is, aside from it being maybe a platytenid or
Not so
benthic
easy. I want to be able to look
ctenophore, is
into
that you
it.
need to see
Yeah, they're also talking about the mouth.
the little
Could you,
lures,
full wide, please?
the teeny, the feeding tubes or filaments, because in the genus Lyroctes, which was mentioned yesterday, it's got branched teeny filaments, and there's another genus, I can't even pronounce it, that doesn't. But in all of the ones we've seen, they're not showing their teeny.
right,
The
let's
teeny
try
are
this
totally
again.
retracted inside those 2 lobes, so we can't
Tilt down.
see them and really nail down the ID.
Okay, so let's see. Watchley pilot.
Go ahead.
Yeah, if you can spend a little bit of time trying to get in there and getting some closer images of this ctenophore might help a little bit with the identification.
Okay.
Yeah, they're being
All right.
extremely
Yep.
uncooperative.
Okay.
Well, part of it is because
If were out, we would see them fairly easily. They're not small structures. They're very long strings that emerge from the tip of those lobes.
you think it's worth it, though, Scott and Chris, to spend a little bit of time to get some closer images?
I think what I remember.
I think so, because they're not out. We can't see them.
I agree with Chris. I don't think we'll learn
Slight
anything
tilt
more.
down.
I just put something in event log. This does look, with the surface ornamentation, like something that's been collected near Hawaii in the genus Lyroteus.
Okay, great. Thank you.
There was a fish coming right
okay,
at you.
here you go,
Okay,
Bruce.
full wide.
What do we got here?
Beautiful.
Oh,
yeah,
we know what that is. Okay, what is that?
Okay. Zoom on the ctenophore.
Is that an onaga?
Zoom on the ctenophore.
Yes, it is. That's one of the commercially
slight
important
tilt up.
bottomfish species. In fact, it's probably the most sought-after species. It's really appreciated
shot.
here in Hawaii, and I assume also in Japan because of that bright red color. It's called a ruby snapper or a scarlet snapper. They're really popular around New Year's to serve on the table because red
center, very
fish in
center
local
string, slightly
culture
right.
is
You can
really
see
appreciated
the,
on New Year's.
Coiled up.
So that's definitely one of the
Rotate
species
right a
we were
little
looking
bit.
for the bottomfish survey for National Marine Fisheries Service. Chris has spent a lot of time studying these, and he might be able to fill in some more information. I'll put him on the spot here.
Do you have any more zoom roll?
That is Max.
Thank you very much, Bruce. So the Onaga,
Polyps there.
so it's the adults are up in the water column. They feed on little shrimp and crabs and little fish and larvae and all types of things that are up in the water column. Fishermen sort of think that they feed a lot at night, right after dusk or before sunrise. And the juveniles, though, are hugging the bottom until they get to be big enough where they're not so vulnerable to predation themselves. So even though these fish are very voracious predators themselves, they got to be careful early on in life. And then at some point, the smaller ones get comfortable enough to move up into the water column and join the schools up above the substrate. And I don't know, I think that's the more interesting aspects of the fish, except that it's beautiful, beautiful fish. Commands extremely high price around Christmas time. It's here in Hawaii, having Onaga on your plate for Christmas dinner is just the best that you can possibly do.
And in Japanese, Onaga means long tail. So O means tail, and Naga means long. Asako Matsumoto gives us some Japanese lessons every time we've got a common name that has Japanese language origins. Thanks, Asako.
As we're looking at the
one of
ash,
the commercial fish species too that's carefully managed here in the Hawaiian Islands. There are areas to fishing for them, areas that are open. I think there might be, and I should know more about this, but I think there might be quotas set. No, maybe not quotas, but seasons set for them. Certainly size limits set for them. And so the fishery is very carefully managed for Onaga here in the Hawaiian Islands, and I assume also to a degree in the Marianas.
Yeah. May not be alive, but
what do we have there?
Like a
is it dead or alive?
doesn't look too alive.
I don't know. Let's look at it.
Geez, it looks like a bivalve.
Yeah, it is a bivalve for sure. I can't tell. Well, no, it looks like it's.
Oh, there
looks
you
like
go.
it's alive. Yeah. Oh, okay. Could it be a brachiopod? It looks like, no, I think it's alive.
Yeah, it's definitely alive.
Yeah.
We saw the mouth.
Yeah, did you see it? Yeah, okay.
Fiber start to close.
Mm-hmm. Nice.
Oh. As we look at the geology here, there's obvious layering in the ash. The brown ash is probably older, and the darker black looks like it may be draping it, and I'm wondering if that might be from what Mary Jo told us earlier,
layers.
could be the more recent eruptive material. So maybe as we get up a little further up the slope, we'll be able to find something that we can collect that has some fresh glass.
Along that line, Patty, I was wondering how it might not be possible because the ash maybe is so loose, especially the black ash that is maybe relatively fresh. But if we could get one of these plates that are kind of the lithified or semi-lithified ash, that there is another kind of volcanic glass that are contained. They're tiny blebs of glass that are contained in the silicate minerals called olivines that crystallize, and so these ash layers are a good place to look for olivines that contain called melt inclusions.
Okay.
That's interesting. The way that
maybe if the ash it looks like it's continuing in every direction as far as we can see so far.
Okay. Well, I shall ask if we can get a piece. Would you like to try for one here?
All right, pilot, you're at about back to 45 in my camera.
Yes, I mean, specific, if it can be the black ash, and I don't know how well lithified that's going to be, because if the lithification is happening during alteration, maybe the black stuff is very loose and hard to sample.
Yep.
But how's the
I'm
current
not
feel
sure.
now?
Do the pilots have a better sense?
Yeah, I'm afraid that the
Yeah,
black
just as the tether
is
looks a little
just,
more in line now.
is just slightly dusting this
Strange.
older altered ash, except maybe right here in the horizontal layer.
Ooh.
What do you think?
Zoom in on that, Roland?
Pilot,
What
could
is that?
yeah, could, if we zoom
Just
in on the ash, the dark
interface
stuff
there between the ash and the lava. There.
Okay, Mary Jo, what do you think about the exposure that we're seeing here? Does the dark stuff look like it's intact? Because it might be possible, you know, with ash, it's pretty easy to put the one arm of the manipulator into it, kind of just crack it off, because it's quite friable. What do you think? Would you like to try for a sample around here somewhere?
Yeah, it might be worthwhile. I mean, if the pilots are willing to try something like what you just
I mean, like this one, like that kind
and
of.
if it works, I think it's possible the rocks could be interesting.
Okay, Pilot, what I'm thinking, we have had a request to try to get some of this darker ash layer. It's probably pretty easy to crack off, but if you could go just a bit to the right in the dark layer, where more of the dark ash is exposed, and maybe even a little further to the right.
Okay, you're gonna. If you guide me, I will take you there. So that's that ash that we were looking at is too light for this discussion.
Yeah, right around 3 o'clock.
Okay.
And then if we zoom in right now, there is a. Looks like there's sort of a tongue of this sticking out.
Okay.
The lasers are just immediately above and to the right of it.
Okay. Okay. I've got it now.
Yeah, lasers are in the older stuff now, and that's not what we want. Yeah. So I see you positioning.
It's just at the bottom of the screen right now.
Right at the bottom, and lasers are just a little bit above it.
Yep.
So we want the darker stuff slightly to the left of the lasers and down. You might be able to flake off some of that ash.
All right, let me get settled in here, and we'll give that a shot.
Roger that. Thank you very much.
When you flake that off, that's going to go flat and out of the grip of your pincher's curved tips.
Yeah.
Sometimes the top of an ash layer will turn brown and get all altered, or a face that's exposed. But it does look as if maybe this is a portion of the ash deposit that was broken off, and the fresher, darker materials exposed on the inside.
Right?
So fingers crossed, might be able to get some of this.
All right. Should we see if we can get a piece of that?
Absolutely.
Want to zoom in and see what it looks like?
Enabling HPU.
Yeah, go for it.
Yeah, it may be very delicate.
ramped
Might
up.
break off in your manip.
Do you want me to put mini Zeus up?
This looks like a very
that'd be good. Thanks.
visual standpoint. I don't know if this will work, but the pilot's going to give it a shot.
I will rotate over to craft.
I'm guessing if he cracks it off, it's going to get flat, and then the curved
Right. Do you
tips
want
of
to
the
look down with mini
pinchers
Zeus?
are not going to be able to
that'd be
pick it
great.
up.
We look right down at the.
Yeah.
Oops.
There you go.
Understood. Video. And pilot, if you are successful, it might be necessary to put this in a bio box.
Okay. Oh, because it's fragile.
Yeah, because it might break up as we jiggle on the surface when we recover the vehicle. Oh, looks pretty hard, Mary Jo.
That's the surprising. I was expecting it to fall with
Can you repeat that? You're breaking up just a little
Small
bit.
piece just broke loose.
Very small piece. Huh?
Sorry, I was just saying that I was surprised. I was
fingers
sort of
under it.
anticipating it would
Lift
break
it
up
to
into
break
a
it and
million
still have
tiny
maybe
pieces
claw
as soon as they touched it, but it actually looked
grab.
sort of solid.
Yeah, it
I
does.
think the craft just has fat fingers.
Well, actually, some of it has broken off, but as you say, you know, it's
Yeah, pilot.
probably
I know you're aware
no
of
more
it.
than
Rotating hard right.
a centimeter or so thick, this little layer. I'm not sure the pilot's going to be able to grab that with a manipulator.
That's fine. It could be that another opportunity presents itself farther up.
You want to try to look farther up?
I think it might be best. This looks a little bit trickier than I was imagining.
Yeah, okay.
This little,
Pilot, we
point of
figure that
rotation you have.
this might be too difficult to get anything out of. What's your feel for it?
It looks like I can get you a small piece, like smaller, like about the size of a ping pong ball.
Okay, if you think you can, that would be great.
It's also a
Okay.
potential ping pong ball size just above this little layer.
So Mary
Dropped
Jo,
a little
Pilot's going to
pilot.
try to pick up some of this.
yes, pilot. I assume this is going to be
We'll
a
just
firebox
see
item.
if it's possible.
Yeah.
Otherwise,
Probably
we'll go